Hi, today I'll be presenting Camptosar by Pfizer, an anti-cancer drug from plant cell culture from plant alkaloids. I'll first introduce what Camptosar is, then speak about its research and developments, elaborate its upstream and downstream processes, and end with Pfizer's good manufacturing practices and quality assurance of Camptosar. Okay, let's get started. Camptosar is manufactured by Pfizer. Camptosar is the brand and irinotecan is the drug. Irinotecan is a major anti-cancer, antineoplastic, or cytotoxic chemotherapy drug used to treat metastatic colorectal cancer. Peak global sales of more than $1.1 billion since its release in the US market in 1996. Metastatic colorectal cancer means that it is an advanced stage malignancy that spread to other areas of the body, but it originated in the colon or rectum. While colorectal cancer can spread anywhere in the body, it most often affects the liver or lungs. I'll go into detail what exactly colorectal cancer is in the next slide. This medication is classified as a plant alkaloid and topoisomerase I inhibitor, which I'll also explain more in later slides exactly what this means. As seen in figure 3, it is given by infusion through a vein intravenously. There is currently no pill form of this medication available. Doses of irinotecan depends on many factors including your height, weight, general health, and the type of cancer or condition being treated. This drug is a chemical irritant, which means that it causes inflammation of the vein through which it is given. If the drug escapes from the vein, it can cause tissue damage, but thankfully, there are trained nurses and doctors that help make sure that this doesn't happen. And I've also included the common side effects of irinotecan that is experienced in more than 30% of patients in the last bullet point. So what exactly is colorectal cancer? Colorectal is a broad term describing cancers of the colon, rectum, or both. This cancer is the second leading cause of cancer-related death in Europe and is the third most common form of cancer in the US, with roughly 1,360,602 new cases diagnosed annually worldwide. Most colorectal cancers generally develop over time from precancerous polyps. Polyps are tissue growths that often look like small, flat bumps or tiny mushroom-like stalks. Most polyps are small and less than half an inch wide. Colorectal polyps are abnormal small clumps of cells that forms in the lining of the colon or rectum caused by a series of mutations or abnormalities that arise in their cellular DNA. Polyps can be carcinogenic when cell mutations proliferate or reproduce rapidly due to the risk factors listed on this slide that's involved in colorectal cancer. If you see figure 4, it shows us roughly where we can find these colorectal polyps in the human colon and rectum. Processes of Camptosar is protected by drug patents assigned by the US Patent and Trademark Offices. This gives exclusive legal rights to the patent holder, which may include entities such as the drug brand, trademark, product dosage form, ingredient formulation, or manufacturing processes. I've included in this slide 2 patents relevant to how the value of Camptosar processes are protected. In figure 5 is the US patent for the processes of camptothecin synthesis, the main compound in irinotecan, to be used as an anti-cancer treatment in 1984 by the Japanese company Kabushiki Kaisha Yakult Honsha until 2003. While Pfizer is the main manufacturer for Camptosar in the US, processes of camptothecin synthesis is currently patented by Yakult Honsha Company till June 2023, as seen in figure 6. Let's talk about where and when this camptothecin compound was first discovered. In this slide, I've included a timeline of the discovery and development of camptothecin. This natural alkaloid compound can be extracted from the bark and stem portion of the Chinese Tibetan plant Camptotheca acuminata, also called the Chinese happy tree, and act as an anti-tumor agent. It has fruit that looks like miniature branches of bananas and can grow up to 75 feet tall. Camptothecin has long been used in traditional Chinese medicine to treat psoriasis, the common cold, and liver and stomach illnesses since the 1930s. The first documented isolation of camptothecin in Western medicine was not until 1966, where it was found while screening various plants for novel steroid compounds. Shortly after, it was then discovered that this compound was shown to exhibit anti-tumor activities in both in vitro systems and mouse leukemia models. From a chemical standpoint, Camptothecin is poorly soluble and has a low in vivo stability. To overcome these issues, in 1990, structure-activity relationship studies were carried out, leading to the discovery of effective water-soluble semi-synthetic and synthetic derivatives. This discovery led to two main camptothecin derivatives, irinotecan and topotecan. Then, in the year 2000, the search for prodrugs approaches were prepared. The CZ48, also known as 20-O-propionate CPT formulation approaches were attempted through encapsulation. The in vivo formation of insoluble and cytotoxic camptothecin in the plasma generated many issues during clinical trials. To face this problem, from the year 2005 to 2010, several derivatives with ring A substitute, glycosyl phosphatidylsulfate sulfate, and carbamate groups were synthesized and biologically evaluated. This discovery led to conjugation with monoclonal antibodies, glycoconjunction, and poly-L-glutamylation improve solubility in irinotecan aqueous solutions. Metabolic studies have shown that irinotecan is a prodrug which needs a hydrolytic enzymatic system to generate the active derivative SN38, an active metabolite of irinotecan with 1,000 times more activity than irinotecan. Building on research findings from 2015 onwards, several novel pharmaceutical platforms were prepared using CRLX101, a nanoparticle conjugate of a linear cyclostrine-based polymer and camptothecin, to further develop and modify the delivery mode to improve tumor cell uptake and reduce toxicity of irinotecan. Today, the camptothecin compound is extracted from these three main plants in Figure 8. On our top right side is the fetid holly or stinking tree, which is primarily found in the Western Ghats of India. The left bottom side is the Indian snake root mongoose plant, which is also primarily found in the Western Ghats of India. And lastly, on our bottom right side is the Chinese happy tree that I've spoken of earlier. The production of camptothecin relies primarily on plant materials harvested from naturally grown trees, which is causing great environmental concerns for these plants. The yield of camptothecin from field-grown trees vary widely and depend on factors that are difficult to control. For instance, plant diseases such as leaf spots or root rot, which are some of the major fungal diseases that can limit the cultivation of these plants and diminish the production of the compound. To combat this, optimization of in vitro secondary metabolite production can help in commercial production using these four main plant metabolic engineering methods summarized in Figure 9. All parts of the three main plants, but seeds, bark, and young leaves, are currently used for camptothecin extraction. The four main methods include the cultivation of transgenic plant via somatic embryogenesis regeneration, hairy root culture, plant cell suspension culture by fibrio callus culture, and adventitious root culture, which are unique techniques in inducing faster growth and biosynthetic stability. These tissue culture techniques provide continuous, reliable, and renewable source of valuable plant pharmaceuticals. These techniques can be used for a large-scale culture of plant cells from which these secondary metabolites can be extracted to optimize production and yield of camptothecin in large-scale bioreactors without depending on wild resources. In this presentation, we'll concentrate on one of the most studied and scalable approach for in vitro secondary metabolite production, which is the cell suspension culture method. Reasons for this approach is its simplicity, easy to upscale, and availability of reactor designs, as they are nearer to the most established microbial reactors. To scale up and produce secondary metabolites in bioreactors, optimization of cell suspensions at laboratory scale is a primary requisite. One of the developments in cell suspension culture of C. acuminata was by the endophyte Fusarium solani from the strain MTCC 9668. Endophytic fungi have potentials for producing the secondary metabolite same as the host plant. Endophytes are a large group of microorganisms colonizing in the tissue of plants without causing overt systems or apparent injury to the host. The plant is thought to provide nutrients to endophytic fungi, while the microbe may produce factors that protect the host plant from attack by animals, insects, or other microbes. It's been proven that endophytic fungi can be a source to provide secondary metabolites diverse in structure and biologic activity. Therefore, the endophytic fungi in C. acuminata plant are expected to be a potential source for camptothecin. The fungal culture was maintained on potato dextrose agar medium and grew on the solid medium as slants at 28 degrees for 7 days until sporulation. Spores were collected by washing with 5 ml of saline, and solution was filtered through cheesecloth to remove any hyphae. The spore solution is stored at -80 degrees with 16% glycerol stock until it is needed. Suspension culture of F. solani is then initiated by adding the spore inoculum in 50 ml potato dextrose medium. The initial pH of medium is then adjusted to 5.6, and the suspension culture was grown at 28 degrees in an incubator shaker rotating at 120 rpm for a cultivation period of 14 days with enhanced small molecule elicitors. I've included in Figure 10 to give us an overview of how complex metabolic processes of irinotecan work in the human body. Irinotecan in the bloodstream converts camptothecin similarities by carboxylesterase into SN38, an active metabolite. SN38 potency is believed to be responsible for irinotecan cytotoxicity. Stabilized irinotecan compounds contain camptothecin compositions that comprises of camptothecin liposomes. This compound generates antioplastic activity that stops cancer cells from multiplying by inhibiting the topoisomerase 1 enzyme in DNA replication. Topoisomerase 1 modulates the DNA topology and twists DNA into a specific spatial structure during replication and transcription with its nuclear enzymatic activity. The formation of a CPT11 topi DNA complex blocks the DNA replication fork. The single strand of DNA breaks and thereby leads to cell cycle arrest and apoptosis. Studies have shown that the topi expression in cancer cells may be 14 to 16 times higher than of that normal cells surrounding the tumor. Owing to the dose-dependent inhibition of the enzyme, cancer cells are more susceptible to toxicity. Irinotecan has been studied in preclinical trials by Pfizer for the FDA to test its safety and efficacy against tumor size differences and rate of survival in combination with 5-FU and LV and as a single agent. When given as a component of combination agent treatment, irinotecan was either given with a weekly schedule of bolus 5-FU and LV, or with every 2-week schedule of an infusion of 5-FU and LV. First-line therapy in combination with 5-FU and LV, studies 1 and 2 are both seen in Figure 11, as Figure 1 and 2 respectively. They were both Phase 3 randomized control multinational clinical trials to support the use of Camptosar injection as a first-line treatment of patients with metastatic carcinoma of the colon or rectum. In each study, combinations of irinotecan with the 5-FU and LV were compared with 5-FU and LV alone. Study 1 compared combination of irinotecan and the bolus form of 5-FU and LV therapy given weekly with a standard bolus regimen of 5-FU and LV alone given daily for 5 days every 4 weeks in 225 patients, while Study 2 evaluated 2 different methods of administering infusional 5-FU and LV, with or without irinotecan for the same amount of study period with 199 patients. In both studies, the combination of irinotecan, 5-FU, and LV therapy resulted in significant improvements in objective tumor response rates, time to tumor progression, and survival when compared with 5-FU and LV alone. These differences in survival were observed despite secondary line therapy in most patients on both arms, including crossover to irinotecan containing regimens in the control arm. To prove effectiveness of Camptosar on a large non-medical scale, we can look into measured QALYs. QALYs are commonly used in health to economically evaluate the value of health outcomes. Since health is a function of length of life and quality of life, QALY was developed as an attempt to combine the value of these attributes into a single index number. Here we'll discuss 2 separate peer-reviewed articles from the year 2013 and 2022 from the UK and Taiwan. In the study from 2013, it was estimated that cetuximab monotherapy and panitumumab monotherapy studies provide poor value for money versus the hybrid cetuximab and irinotecan study, while in the more recent study from 2022 proved that 75 patients with mCRC, irinotecan dose escalation appeared cost-effective with considerable quality improvements during the study period. But of course, with any study limitations, further randomized multi-institutional controlled trials are warranted to validate the results from both studies. To better understanding of the effectiveness of Camptosar, future quality studies could combine the two study methods and research hybrid dose escalation therapy of irinotecan and cetuximab and/or panitumumab. The optimal upstream processing choice to optimize camptothecin yield for production of irinotecan is currently by using semi-synthetic processes through the Endophyte F. Solani MTCC 9668, as explained in earlier slides. In this slide, I've included an example of an industrial-scale fermentation production system that can implement findings from Venugopalan AL's findings. Their findings showed that by improving the fermentation parameters of pH, temperature, and agitation speed on the biomass, camptothecin production was enhanced by around 152-fold from 0.9 to 0.28 micrograms per liter per day. To further improve this yield, various elicitors and precursors such as tryptamine and bovine serum albumin during the cultivation period optimized and improved the yield of camptothecin compound of up to 4.5 and 3.5 fold respectively. I've included figure 12, which is a simplified flow diagram of how these upstream processes are achieved through media hydration, filtration, cell culture bioreactor, and harvest collection before the start of the downstream processings. Following the upstream processing, further downstream processing is needed to develop irinotecan through the remote loading and passive loading method to separate and purify the compound. These optimal downstream processing parameters were adapted from Yoshino al., who are the current patent owners of the preparation of irinotecan used in Camptosar. Figure 13 is a simplified flow diagram of how these processes are achieved through separation, purification, finishing, bulk storage, and finally filling of irinotecan. To guarantee quality assurance of Camptosar, Pfizer is bound to practice GMP, Good Manufacturing Practices, that aligns with the FDA. This is a system that consists of processes, procedures, and documentation that ensures manufacturing products of pharmaceutical goods are consistently produced and controlled according to the set of quality standards. This includes post-marketing requirements and commitments that refer to studies and clinical trials that the sponsors, like the FDA, conduct after approval to gather additional information about the product safety, efficacy, or optimal use. The following adverse reactions have been identified during post-approval use of Camptosar. Because these reactions are reported voluntarily from a population of uncertain size, it is not always possible to reliably estimate their frequency or establish a causal relationship to drug exposure. Myocardial ischemic and thromboembolic events have been observed in patients following Camptosar therapy and while receiving Camptosar. Because irinotecan has anticholinesterase activity, it may also prolong the neuromuscular blocking effects of succinylmethonium and the neuromuscular blockade of non-depolarizing drugs. These disclaimers are all available to the public and medical professionals before prescribing Camptosar. Camptosar injections are available in single-dose brown glass vials in package sizes of 2, 5, and 15 ml in single-dose amber-colored polypropylene cytosafe vials. They are recommended to be stored at controlled room temperature of 15 to 30 degrees Celsius away from light and should be kept in the vial in the carton until the time of use. Pro-drug monitoring since the drug discovery should include all these precautions and warnings advised by the FDA. Post-drug monitoring of all these symptoms should generally be observed after 24 hours after drug administration. In severe cases, if multiple symptoms persist, please contact your local medical care professional. And that is all I have for you today. Thank you for paying attention throughout this whole video. Until next time, bye!